there, we're going to talk about the cell cycle and cancer today. So the first thing we need to know about the cell cycle is that cells do not spend all of their time dividing. In fact, they spend most of their time in what's called interphase. And during this phase, the cell is growing and DNA replication occurs. So interphase is where a cell spends about 90% of its time. And we can see, right, interphase from starting here, going all the way around. Right, so if we think of this like a piece of the pie, right, that's a huge chunk of the pie. So interphase takes up a majority of a cell's lifespan. And then within interphase, there are three parts. We have G1, where the cell grows a lot. So G1, G stands for growth. So it's kind of like that first growth phase. And then after G1, there's S. The S stands for synthesis. If I synthesize something, that means to make. So DNA synthesis, right? We are making a copy of DNA. So this is when DNA replication occurs. And then, of course, there's the final stage, which is G2. And then this is where the cell grows some more. These pieces of the pie are also representative of how much time is spent in G1, S, and G2. So we know interphase is 90% of the cell's life. And of that, most of that time is going to be spent in G1, where a cell is just hanging out. It's doing its everyday business of being a skin cell or being a nerve cell. And then if it gets a signal, it'll head to S phase where DNA replication will occur, and then G2, and then it will head into what's called mitosis, right, where we actually get a second cell. So mitosis is when the nucleus divides, right? You start with one cell and you get two. But mitosis is just division of the nucleus. In order to actually get two daughter cells, we have to have what's called cytokinesis. And this is, again, just a fancy way of saying that the cell divides into two, or division of the cytoplasm. So mitosis is when the nucleus divides. Cytokinesis, we could say, is division of the cytoplasm. And then both of these together makes cell division. So as far as the cell cycle, there's two stages. Interphase, which is G1, S, and G2, and then cell division, which is mitosis and cytokinesis, where we divide the nucleus and then the actual cytoplasm to end up with two daughter cells. A quick comment about cytokinesis. In animal cells, because there's no cell wall, the cytoplasm just simply pinches off from one another, and then it just spreads and spreads and spreads until eventually it kind of snaps, and then you end up with, two individual cells. If you've ever eaten Laffy Taffy, you can stretch it, right? And then at some point you stretch it so thin that it pinches off. This pinching of the cytoplasm in an animal cell is called a cleavage furrow. Now, plant cells, right? Plant cells do have a cell wall, so we can't simply pinch a cell wall, right? It's very sturdy, it's very thick. So instead, a plant cell will form what's called a cell plate between the two new nuclei that have formed from mitosis, right? they'll form a cell plate. And again, yeah, it's kind of like you're slowly building up a wall, right? One brick at a time until finally you seal off one room from another. So, and once the cell plate has been formed all the way, it would just be the new cell wall. But while it's forming, we call it a cell plate. And that's only in plant cells because they have a cell wall. Animal cells use that cleavage furrow or that pinching to cleave the cytoplasm apart. So taking a look at cancer, we're going to define cancer as uncontrolled cell growth. Uncontrolled or unregulated cell growth. So our cells receive signals that tell them when to leave G1, right, where they're just growing and having everyday life. They receive a signal that tells them, hey, it's time for cell division. We need to make new cells. And then they'll enter SG1 and then finally cell division. Cancer is when those cells ignore those signals and they just start dividing uncontrollably. So it's uncontrolled cell growth. A group of cells that start dividing uncontrollably are called tumors. And then you can have benign tumors or you can have malignant tumors, right? Benign tumor, meaning that it kind of stays where it's at. It's not cancerous. Or a malignant tumor, which we would say is a cancerous tumor, and you would need to go on to cancer therapies like radiation or chemotherapy or maybe even surgery to help get rid of that malignant tumor because it is dividing uncontrollably and it's causing problems, which we call cancer. We do have proteins that control cell division that tell our cells when to divide and when not to divide. Those proteins are called cyclins. And so we have a built-in system that controls when our cells divide or not. Otherwise, we'd have skin cells, dividing all the time. We don't need that, right? We only need new skin cells to replace damaged ones. Maybe you got a paper cut, or maybe you scraped your knee on the sidewalk and you need to replace the skin cells. But if the cyclins, if the proteins stop working, then you get uncontrolled cell growth. So that's our notes for this page. Let's take a look at our next set of notes. So still talking about cell cycle and cell growth. Move this out of the way. So I've got an illustration here of the cell cycle. So we need to label the different stages, G1, G2 and S, mitosis and cytokinesis, and then describe what happens during each phase in the division of the cell. Okay, so they've already given us interphase. So I'm just going to kind of block that off. All right, so this is interphase. And then grab another color. So this would be cell division. There we go. Okay, so cells start in interphase. Specifically, they start in G1, which is that first growth. So this is when a cell, let me actually come up here. So this is when a cell is normal, right? It's just doing normal, everyday functions, right? Protein synthesis, maintaining homeostasis. So the cell grows and will build proteins needed for cell division. Right? So it's normal everyday functioning, but it is still preparing for when we move on to the next part, which is S, right? Or synthesis. In the S phase, Right? We have DNA replication. Individual chromosomes and chromatids are hard to see at this phase because they're still in their chromatin form, but the cell will condense the chromatin into chromosomes so that we get two copies. So we'll go from a single chromosome to a double chromosome. This way, when cell division occurs, we don't lose any of our chromosomes. And then finally, there's G2. This is when the cell will grow a little bit more. So grows and takes in nutrients to get ready to divide. And then this is where we'll see a very special organelle called centrioles. Centrioles are used in cell division to help actually move the chromosomes so that we get the correct number of chromosomes in each daughter cell. So that organelle is the first time we're seeing it. Then we move finally into the M phase, right? Mitosis, where we have prophase, metaphase, anaphase, and telophase. And then last but not least is cytokinesis, right? C. So mitosis and cytokinesis together gives me cell division. So I start with one cell, right? One parent cell here in the beginning. There's my little nucleus. And then as I move through interphase and cell division, I will end with two daughter cells that are identical to each other as far as the DNA is concerned. Okay, I think that's everything we need to know for our cell cycle and cell growth, right? Interphase, made up of three parts, G1, S, and G2. Cell division, made up of mitosis and cytokinesis. If you have any questions or concerns about the cell cycle, please let me know.